Can everyone see this, the metabolomics and exposomics slide?
Yes.
Okay, so for any resources, you can just jump down to the little website at the very bottom of the page here and click that. That has our laboratory website, and you can get a lot of information from there. So my disclosures are that I'm an unpaid scientific advisor for 3 groups, and I also receive research funding from a company developing serum to treat autism spectrum disorders. So the background literature, I've highlighted the PMIDs in Cayenne, so you can quickly look at these. But basically, these are some of my published papers that tie into this work. And this, since it's being recorded, you'll be able to take a look at any of these that you like. So the outline of my talk starts, the first half of the slides are going to be talking about the why we do what we do. And the second half is the what we found, okay? And so we'll talk about the philosophical foundations of a new medical perspective of chronic illness, regeneration, and healing. We'll talk about the cell danger response, the healing and aging cycle. And then we'll talk about some of our results from metabolomics and exposomics, and then the convergent lessons that are emerging from 10 clinical metabolomic studies we've completed. And then I'll end with some statistical caveats, from the battleground of big data analysis. Okay, to start out our philosophical section, I start with a quote by John Dryden in a poem from 1681 that self-defense is nature's oldest law. So we've all received from our ancestors all the genes that they needed to survive every pandemic, every famine, every hard winter, at least until they had their first child. So those are the genes that we've inherited, okay? But the environment's changed. And so, those genes are not enough. Another quote that I like is from Nelson Mandela, and he says that when a deep injury is done to us, we can never heal until we forgive. And then I was talking to Keith last night, and he asked this similar question: how does a cell decide when to forgive? Obviously, that's a metaphor, and begin to heal. And so the answer is a cell will only redirect resources, and those resources are both energetic and material resources, so matter and energy and information, away from cellular defense toward healing when it receives the signals that say the future is safe. And the corollary is that regeneration and healing can't proceed until persistent triggers of the cell danger response are removed. Okay, like if you keep ripping the scab off of an abrasion, it's going to take you much longer to heal, and you're going to have scarring. Okay, if that keeps happening. So then this big question, overarching question: why do people become chronically ill? Well, I believe that that's because they have encountered a perfect storm of usually dozens of environmental and genetic factors that produce or persistently reactivate an injury and then block the normal cycle of healing. And the corollary is that all chronic illness represents a failure of our ancestral genes and our inherited epigenetic modifications to adequately manage the response to environmental conditions that we've experienced. And so basically it leads to this logical tautology. People are sick because they can't get better, okay? So why can't they get better? Well, I believe it's that the information is in the molecular analysis of the healing process itself. So the organizing principles of our research start out with this one fundamental fact: injury is universal. Living cells try to heal themselves, and cells that can't heal cannot transmit their DNA to the next generation. So they don't, and they die, and those genes, that gene set dies out. Chronic illnesses are the result of a mismatch between genes, environment, and the developmental timing of the injury exposure. So gene by gene by environment by timing, or we refer to this as ecogenetics. And then finally, the last organizing principle that Chauvin has iconized, and that is metabolism is the real-time interaction of genes and environment. So our genes give us a certain plasticity to respond to environmental insults, and so metabolism lives in the moment. And I'll follow this up by saying metabolism is fractal. There are it's like an ancient oak tree where there are metabolites that are trunk metabolites that don't change at all. There are very thick branches, and then there are leaves that flutter in the wind, and even those can be remarkably useful in understanding disease. For example, one of the metabolites that flutters in the wind like a leaf is glucose. But we know that fasting glucose can be very useful in looking at someone's insulin resistance or sensitivity. Okay, so that's for you, Chauvin, down at the bottom right. Okay, so the take-home messages are that pathogenesis does not equal salogenesis, and I'll define that word, but it's basically the process of injury does not equal the process of healing. This is really fundamental and absolutely a missing piece of medical approach to disease that we all were trained in. So the cell danger response and mitochondria coordinate both of these processes, and metabolomics can be used for both pre-symptomatic risk assessment, triage diagnosis, and prognosis. Okay, so where does this leave us? So we cannot see what we cannot imagine. Well, I won't go into this, will take too long if I go into too many analogies, but I'll say our training creates subliminal limits or unrecognized horizons in our vision so that, new tools and models, can be used to extend our scientific vision. So sometimes in order to see, a new world, all you need is to create a new lens or a microscope or a telescope or a mass spectrometer. So a paradigm shift is needed to understand chronic illness. And I'll say this many times throughout the talk, acute disease is not the same as chronic disease. You cannot use the same principles and hope to get the same results. So I consider acute disease the last the medicine in book 1 is the last 5,000 years of recorded human history, where we really focused on managing what happens if you break a bone, trauma, or poisoning, or if you have infections. So medicine's gotten good at this. But book 2 of medicine is really anything that persists for longer than 6 months. Okay. All right, so imagine a vibrantly healthy human as, this house on the left before disease. Well, you can suffer injury, and there are mechanisms of injury. In this case, it's a fire. That is my metaphor for pneumococcal pneumonia or a viral encephalitis. Okay, we are trained to identify the mechanisms of injury and then to treat those. Okay, so we can treat that fire, but then what we're left with is a house that is destroyed. Now, all of first book medicine stops with step 3 and then relies on spontaneous healing to occur. We do this so subconsciously that it's automatic that if we give, let's say penicillin for a pneumococcal pneumonia, we just assume that all the pulmonary, destruction that has occurred as part of the infection will heal itself. Okay, and we forget that's an active process. So rebuilding requires active healing, or what I call salogenesis. And that is what I believe is a critically missing component of modern medicine. So Salus was the Roman goddess of personal health, welfare, and safety. This slide starts with basically saying that healing is not the reverse of pathogenesis. So we can start with health. You can suffer a traumatic injury, be exposed to toxic metals or pollution or viruses, tobacco, infections of different kinds, and get over to the right square, this red square, which is a chronic illness. But it turns out, so we can study that. And what first book medicine teaches us is we just eliminate those problems and everything will get better. Okay, that's what first book, but that's not how living systems work. That's how you repair a damaged or imperfect Hubble telescope, is you analyze its optical properties, you create a fix, you fly it up, install it, and then you enjoy the fruits of your engineering fix. That's not how cells repair themselves, okay? So there's a beginning and a middle and an end to this process of returning back to health that requires transformation of mitochondria from different developmental phenotypes, what I call M1, M0, and M2 mitochondria, okay? So in this first book of medicine, treatments of chronic illnesses will lead to palliation. So they're usually symptomatically based. So if someone has a headache, you can give them an aspirin, or you can, remove the hammer that keeps hitting them. Okay, so based on the word salus, and we've used this word salogenesis to describe the molecular and cellular events that are necessary for going from a state of injury to a state of health. And that's what I call second book medicine. And what I believe that treatments that really focus on this second book on healing actually leverage the internal ability of cells to use information is stored in our DNA and as well as energy produced by our cells and material resources in the form of amino acids and nucleotides and fatty acids and sugars. Okay. So I gave this talk in April of last year at NIH, and the organizer that invited me, said, "Great talk, Dr. Naviaux, but I hope you're wrong." And I said, "Well, why is that?" Well, if you're right, it means that the National Institutes of Health has been funding only the first limb of a two-limb problem. We've only really been funding, understanding the pathogenesis of illness and not the salogenesis. So for 70 years almost, the NIH has really focused on building deep silos of knowledge for many, many different diseases, for primary mitochondrial disease, autism, chronic fatigue syndrome, Gulf War. I put these silos here because these are the silos, 10 of these are ones that we've already studied, and ALS, Parkinson, and Alzheimer are ones that we will be studying over the next few years. And so what people, by building these deep silos of knowledge, have done is they have focused on the differences between illnesses. And what we are focusing on is the similarities, this hidden, the commonality that all diseases share is in order to get back to health, there is an underlying root to the process of recovery. So the things that make chronic diseases the same are more important for healing than their differences. So this has, political and social implications for our unrest in America at the moment. But really, it's our similarities that are more important for healing than our differences. So how did I get into this? Well, I got into it because of patients with primary forms of mitochondrial disease, genetic forms of mitochondrial disease. So these are diseases called MELAS and NARP and Alpers syndrome. We actually discovered the gene that causes causes Alpers syndrome, which is the oldest Mendelian form of mitochondrial disease, first described in 1931. But anyway, so under health conditions, mitochondria are a reticulum. They are almost a root system that go throughout the cytoplasm of a cell. So here are two HeLa cells next to one another down at the bottom with an interconnected network of mitochondria. But when there's any kind of injury, mitochondria fragment and they change their metabolism and they become pro-inflammatory. Okay, so these are anti-inflammatory organelles that are consuming oxygen. These are pro-inflammatory organelles that are no longer consuming oxygen efficiently. When health is restored, the mitochondrial reticulum is restored. They're reconnected. But when that's blocked, they remain disconnected. This disconnection has been seen also even on the space station. When you grow cells in microgravity, mitochondrial would become disconnected. So anyway, when this happens, when you're blocked to healing, 60% of adults in the world today suffer with at least one chronic illness, and actually 40% of children, in their up through their teens, suffer with at least one chronic illness. So we've conducted over a dozen different studies, 2 in autism and 2 in chronic fatigue syndrome, but also Gulf War illness and post-traumatic stress disorder, traumatic brain injury, recurrent major depressive disorder with and without suicidal ideation, an autoimmune liver disease called primary sclerosing cholangitis. We've studied exercise and aging and these primary mitochondrial diseases, and then a longevity default pathway in C. elegans called Dower, and some breast milk biomarkers that I'll talk about. Well, where does that all lead us? It leads us to what we call the cell danger response. So the core pathways of the cell danger response are regulated in over a dozen chronic illnesses that we've studied. And so if you think of measuring metabolites, a set of metabolites that are unique for a particular disease, as well as metabolites that are shared and pathways specifically that are shared by different diseases, what you come up with is something that looks like a sunflower, where the shared pathways are the pathways of the cell danger response. And those involve purines and pyrimidines, phospholipids, acylcarnitines, fatty acids, sphingolipids, eicosanoids and endocannabinoids, cholesterol, folate, B12 metabolism. I can go through it, but basically all these are part of a system of cogs and wheels that are coordinately regulated. These are not things that are activated in isolation. They do not work in isolation. They work as an integrated system. So what is the cell danger response? So here's a cartoon of a cell. An archetypal stress was a viral infection. And one of the first things that happens is, I'll point your attention to number 4 over here, is actually a conversion of filamentous mitochondria to segmented mitochondria. We call this the spaghetti to meatball transformation. Okay, so this is, and that's actually not just a joke, it's what the mitochondriacs call it. Okay, so the spaghetti to meatball transformation. But when that happens, then the intracellular dissolved oxygen concentration rises. When that happens, then NADPH oxidases are activated and superoxide and hydrogen peroxide are created, and you get a host of many other things. So this is an ontogenetic response, meaning that it's a developmental as well as genetic response initiated by what I call electron steals, because mitochondria regulate their metabolism according to the electrons they receive from metabolism. When those electrons are stolen by a virus, let's say, so to make its own proteins, its own lipids for cell envelopes, or nucleic acids, then there's fewer of those that are available to mitochondria, and that results in this filamentous to fragmented morphological transformation. And so I'll refer you to the paper. We can go through, I won't go through everything here, but the important thing is that this is a coordinated response, but people tend to look at this through a keyhole of their own research interests. So some people might just look at sphingolipids. Other people might look at cholesterol or fatty acids. Some people might look at, demethylation of DNA. Some people might look at endogenous retroviral sequence activation, okay? These are all coordinately regulated responses. All right, so again, to nail this home, so we start in a condition of health. This is first book of medicine, acute illness. We have an injury. Doctors will look after the airway, breathing, and circulation. You do first aid, you provide symptomatic treatment, and because this black box is intact, most of the time people get better. But if the black box of healing is not, getting better, then it tips us into this repeating loop of chronic illness. Okay, so now if we just start with this as a dysfunction as a problem, as a starting assumption in all chronic illness that results from a variety of different problems that are relevant either for children in psychosocial deprivation states or rewounding in adults, it'll pull us into this repeating loop. Okay. But if we unpack that black box and start analyzing it with the tools of systems biology, we can begin to see that there's actually an organized sequence of events that happen. And I call these the cell danger response 1, 2, and 3. And we'll go through those a little later. But if you unblock that, then healing is restored and health can be restored. So this is such an ancient response. Plants have it too. So this is a Arabidopsis. This is a little cricket bite over here. Turns out if you just add glutamate or ATP to this, this genetically modified Arabidopsis that will fluoresce green in the presence of uncaged calcium, this is what happens. Okay. So everyone see that? So that's the plant within 90 seconds transmits the injury that it receives in one leaf to all parts of the plant. Okay. Humans are even better at doing this. Okay, so we spend most of our life in this health cycle of wakeful activity and nutrient intake. That activity releases a little bit of extracellular ATP that then has to get metabolized to adenosine, which is necessary for the induction of sleep. If you don't have adenosine, you can't have normal restorative sleep. Okay. Once that's metabolized, then you can wake up again. So, if the injury is very significant, let's say with a viral infection and there's been cell loss or excitotoxicity, cell loss, then the first stage of the cell danger response is activated. That's inflammation. It's like trying to seal off the bulkheads of a submarine that has been injured so that the rest of the submarine will not be destroyed when one compartment becomes filled. So that's what this containment and innate immunity is all about. And there's a very specific type of metabolism called glycolysis that is used during that time. Let's see. So, and this can be activated by any number of different things. And so I have a stimulated emission video of mitochondria after they've sustained a stress. And within 5 minutes, they transform from the spaghetti to meatball configuration. That changes them from antioxidative, basically anti-inflammatory to pro-inflammatory organelles. All right. So these spherical mitochondria we call the M1 mitochondria. M0s are pluripotential. They can actually be both differentiate into M1 or M2 mitochondria. And M2 are the more filamentous oxphos dependent forms of mitochondria. Okay. So these are just the very specific stages of metabolism that always exist. It doesn't matter what the injury is, whether it's a broken leg, a cut, a scrape, a myocardial infarction, or a stroke. So now is the second half of the talk. So metabolomics and exposomics, when we draw a blood sample, we think of it as a little microcosm of all the resources that all the cells in our body, both the bacterial cells as well as the human cells, need for growth and repair, as well as all their waste products. And we can analyze those, by a $500,000 machine here, a triple quadruple mass spectrometer for metabolomics. And for exposomics, we can look at environmental toxicants using gas chromatography, triple quadruple mass spectrometry. And the workflow is basically take a sample, add internal standards that are C13 or N15 or deuterated standards, run them through an HPLC, run them through the mass spec, isolate and fragment the ions, and then put them through a statistical analysis in order to come up with pathway changes that are reflective of the underlying biochemistry of the illness. So at UCSD we have, so this is an illustration of the kinds of molecules that we interrogate by GCMS and LCMS on the exposomic side. So 1,232. And then we have on the endogenous side, 690 molecules that we interrogate from over 63 different biochemical pathways. So overall, that's 1,922 chemicals that we measure. So this is an example of the receiver operator characteristic curves for 10 different studies that we've done. Each one of these represents a $500,000 of research by 5 different people in our lab. And you can see that using metabolomics produces ROC curve accuracies of about 0.85 up to 1.0 in experimental models like C. elegans. But even in depression, where we're looking at cerebral spinal fluid or plasma, the accuracy of diagnosis is still very high. But what we found through these studies is that the actual prognostic value of metabolomics is even stronger than its diagnostic. So you have to imagine that when you take a blood sample from someone, you are really getting a snapshot of a moving train. Every person is on a train that is taking them to a particular location. We'd like that location to be, one of vibrant health, but people with chronic illness will be on a train that takes them into the future with multiple organ morbidities and typically exaggerated symptoms. So in this study of men and women with recurrent major depressive disorder, we took a sample at time 0, and then we followed them into the future and determined how long it took them before they had a recurrence of their depressive symptoms. And for women, it was about 600 days, and for men, it was about 300 days in this study. But when we use metabolomics to begin to see if we could predict which person would relapse and which would have sustained absence of symptoms, we found that there were dozens of metabolites, and I'll give you just one, is methylcysteine. If we just stratified the patients into those who had the highest level of methylcysteine in the blue down here, and those that had the lowest levels of methylcysteine, it allowed us to tell so the median time to relapse was 400 days here in the orange for those who had low levels, and those who had high levels had actually over 850 days of symptom-free recovery. Same thing for males. We can do the same thing for males, with area under the curve for ROC curve accuracy that over 90%. We've also looked at, we're interested in newborn screening and autism. And so, we not only are doing work with newborn dried blood spots that are taken at the time of birth of every child in the United States, but we are also looking at the breast milk, because the chemistry of breast milk changes according to, well, just to start out, turns out that, we can take a sample of any breast milk and tell you whether that breast milk is being made for a baby girl or for a baby boy. So the milk chemistry changes according to the needs of the child. Okay, and so in this study, we took frozen breast milk samples at 3 different times, then followed the children for 3 years, and then went back in time. So we began looking at the children who had critical neurodevelopmental risks, and went back in time to their breast milk samples. And what we found is that with 8 biomarkers in the milk, we could identify with 91% accuracy the children, in this case 23 children at critical risk, with from 40 neurodevelopmental normal controls. Right. All right, so the last part of this is,
Can I interject there for 1 second?
Sure.
Yeah, I just wanted to ask you, to which extent you think this correlation, there's any form of causality, or it's just something that, because these kids have that problem, impact? What is your interpretation of this correlation? What is causing?
Well, so the information is actually in the details over on the left here. It turns out that cerebrosides, basically these lipids that are of critical importance for developing brains, are fortified in the milk of mothers who have children who are struggling neurodevelopmentally. So however the biological connection is made between mother and child, through smell and sight and scent and touch, the mother's milk is fortified with, basically these dihexacylceramides, a variety of different phospholipids, monohexacylceramides. These are glycosphingolipids that are cerebrosides that are necessary for brain development of a child. So the mother whose child is struggling the most makes the most.
Mm-hmm. So you would predict that a lack of those cerebrosides would actually directly, potentially be causative of the disorder?
No, I don't think of it that way. I always think of everything as a reactionism. Okay, so
so it's an expression of the change in the homeostasis, if you wish, or anything like that.
Yeah. So so I always see this, as the body's response to a problem. Okay, and so the interesting thing is that the milk for neurodevelopmentally normal children had lower levels of these molecules. Just meaning that normal kids, that on a normal course, don't need as much cerebrosides in order to have a normal brain. But the kids that are having problems, are helped by, this maternal child chemical bond.
Thank you.
Okay. So this is a big data question. Basically, if the number of samples N is much less than the number of parameters measured, then you can run into problems that frequently are, not obvious. So we performed a little simulation study where we used a random number generator to populate a matrix of 691 metabolites in 60 patient samples, so 30 cases, and then just basically just randomly divided those 60 samples into 2, so controls and cases. And lo and behold, when we use multivariate statistical analysis, so, basically whether you use PCA or partially square-screwed analysis, you get similar results. Basically, you can always find metabolites that are discriminating between the 2 groups. I've named them ASD and TD here, but really they're just, cases and controls. But the thing that there's already there's a caveat here for anybody who knows metabolism, is that these metabolites come from many different pathways that they're randomly selected from different pathways, okay? There's not an intensification or hierarchical structure to these results. And if we just re-randomize the same data set, we get a completely different set of metabolites. And if we look at, let's say we thought that first was going to be a discovery set and that second one might be a validation set from a different, clinical study one study might be done in Philadelphia, another might be done in San Diego. When you look at this, lo and behold, you get 12 metabolites that are shared by these, and remember, these are random data sets, okay? And in the literature, a lot of people would say, okay, that's what's shared. That's what's important, is these 12 things, okay? Well, wrong. Now I'm going to give you a kind of a quick trick that we use in order to tell the difference between strong studies and weak studies. If you just take, let's say we have 691 metabolites, and we can take the p-values, and that can be, a non-parametric p-value would be a Mann-Whitney U-test p-value, and then you could use a student's t or a Welch's which is what you would choose if the distribution is still symmetric, but the variances are different between the groups. But anyway, if you take all those p-values and you just do a histogram, frequency histogram, you'll see it's roughly equivalent, from the lowest values to the highest values. And in fact, what's interesting is that it comes up, so Q is just a Bayesian false discovery rate. If you take 0.05 times the number of metabolites, you get about 35, which is the average of all these little columns, bars, okay? So this is a weak study that is generated from random data. It's very easy to do too. I mean, this is a very easy thing to rapidly assess the quality of data from big data sets. Now, here's an example of a couple strong metabolomic studies. So here's a mouse study. Turns out it was only 15 animals, 7 controls, and 8, animals that happened to be injected with ATP. But you can see that there is a very strong, increase toward the low p-values, okay? And in a human study, you can see also that there's a very strong increase toward the low p-values, okay? This is really very dramatic and easy to do to rapidly assess the quality of any, big data set.
Is that a description of all pairwise interactions that you could have within the data set, every interaction among all of the 691 or whatever the case may be?
It's related, but that gets into Pearson and Spearman matrices. So it turns out you can also determine, p-values for every pairwise analysis in a Pearson matrix or a Spearman matrix, and do exactly the same thing, and you'll get the same results. So it's very, very useful, quick test, okay? So now if you do we like random forest because it's very robust or resistant to overfitting your model. And in this example, which was generated from a random data set, the out-of-the-box error was 0.6, and we like to see out-of-the-box errors of below 0.25, okay? I won't go through a lot of this. This is just another example. Again, this particular out-of-the-box error was again 0.6, and this is, again, we want to be below 0.25. So now if you have a strong data set, this is what you get. So the out-of-the-box error, the misclassification was 0. So in these animals, okay? And in this particular one, which is another strong data set from major depressive disorder, it was 0.17. So again, below our 0.25, optimal threshold. You can do, heat maps, and you can make yourself believe that, some random data set is going to be different, between cases and controls. But, this is what on the right is what a real, strong data set heat map looks like, okay? With metabolites that are strongly increased and strongly decreased by treatment versus control. So that's it. And I'll take questions. So